Your cells possess a highly restricted protocol that commands them to physically fuse their architecture into massive biological dreadnoughts when standard parameters fail. When an indestructible foreign asset bypasses your outer armor and resists standard enzymatic breakdown, individual scavenger cells recognize they lack the kinetic capacity to neutralize it. Instead of initiating systemic stasis, these macrophages execute a localized structural merge. They intentionally dissolve their outer lipid boundaries and combine their genetic payload, forming a single, gigantic biomechanical entity with dozens of shared nuclei. This newly formed macrocell acts as a high-yield chemical reactor. It aggressively seals itself to the foreign hardware and releases a highly concentrated, extreme low pH saturation directly onto the target, slowly forcing the invader to dissociate its tissue strands. Would you authorize a synthetic enzymatic trigger to permanently lock your immune grid into this macrocellular state, granting you immunity to indestructible biological threats? Once the mechanical failure of the foreign asset is confirmed, this massive cellular dreadnought gracefully dismantles itself, recycling its structural components. We are discovering that human anatomy can dynamically reshape its own scale. It means your cells possess a highly restricted protocol that commands them to physically fuse their architecture into massive biological dreadnoughts when standard parameters fail.